that is on machine learning techniques in drug and vaccine design. Before going to discussion of 13th chapter, machine learning techniques in drug and vaccine design, we recap the last session contents. The last session content is we completed up to the vaccine design, isn't it? What especially on B cell epitope. So, can someone tell me the difference between T cell epitope? Some two or the three differences: T cell epitope and B cell epitope. Thank you, thank you. And even the feedback, whatever I got, I think definitely Shushmita madam is a PG scholar. Okay, so PG or PhD or even a faculty. I don't know about the UG students. Okay, so okay, good. Thank you. May know about Sushmita madam. What are you doing, madam?
Actually, I'm a faculty of in assistant professor in microbiology, sir.
Good. Thank you. Thank
Yeah.
you for your feedback, madam.
Yes, sir. Actually, we are having this in our curriculum, the topic that you are dealing.
Mhm.
Really, it is helping us a lot to
Good.
improve our skills and everything. I'm just
Mhm.
noting down, I'm following the
Mhm.
videos recorded, and it's
Mhm.
very informative session, sir.
Yeah, so one day
Our-
it's not restricted up to the cancer. Some often I was bringing the virus. I'm telling about the SARS-CoV-2 Nipah virus. I know even some many microbiologists are there. So I just integrated this. Good.
Good,
Thank you.
sir. It is very much integrated and it is with interdisciplinary approach is also there. Yes, thank you very much.
Thank you, madam. So let us discuss about the B cell epitope. The first question I asked everyone is about what are the differences of T cell epitope and B cell epitope. What do you understand from this? T cell epitope, B cell epitope. So can I query with, or I can provide you one more hint on this? So what is APC? Actually, from T cell, B cell, there are two types of cell I showed, isn't it? So one cell is antigen presenting cell. Which is antigen presenting cell? T cell or B cell?
B cell.
Good.
B
The
cell.
answer is B cell. B cell is correct. Okay. So other antigen presenting cell examples are very good. Even I appreciate the participant who has answered. Okay. So this kind of interaction is important. He won't forget at all. I think now just because he has answered. So B cell is an antigen presenting cell. Okay. And like for B cell, I want dendritic cell and macrophages. Okay. These cells are dendritic cell, macrophage, B cells or antigen presenting cells. So the only cell which is interacting with the antigen presenting cell is the T cell. The T cell interacting with any of these three cells, not only these three cells, we also have natural killer cell. Okay, we have other cells also. So generally, this T cell is going to interact with all these antigen presenting cells and try to get activated because of signaling. Okay, there are three important signaling. So you can also study more about what are the most important signaling, which gives active the T cell activated. And how about the APC? If for the T cell getting activated, what are the signaling? What are the signal transduction is important, and how about for dendritic cell? Okay, even there are stages of dendritic cell development, and even macrophage or B cell, whatever it is. Oh, good. So we have completed one point. What is APC? The second point is what are the types of T cell epitopes? And similarly, the next question after this, I will be asking you, what are the types of B cell epitopes? Types of T cell epitopes and types of B cell epitopes. You forgot that, isn't it? Cytotoxic T cell, helper T cell epitopes, good. So CD4 and CD8, you remember? So CD4 plus T cell, CD8 plus T cell. So based on these two types of T cells, we have found two types of epitopes. Good, madam. Your answer is right. The next is types of B cell epitopes. B cell epitopes, you have sequence epitopes, that is linear epitopes. The other is discontinuous epitope or conformational epitope. Linear, I type it. Linear is the first one. Okay. And the second one is the, yes, conformational or discontinuous epitopes. Okay, two types of epitopes are there in B cell. Good. So we have completed even the types of T cell and B cell epitopes. If I ask you to give any two tools for T cell as well as B cell, what tools you can show? What are the mention me any two tools which we can use for prediction of T cell epitopes? Plenty of softwares are there, isn't it? So if you search in Google Scholar about immunoinformatics, just type immunoinformatics, you get plenty of hundreds of softwares are mentioned in a single paper. Discotope, very good. So actually, Discotope is a kind of the structure. You have to feed the peptide structure to find this. Very good, ELIPRO. Okay, Discotope, ELIPRO. And the other famous T cell epitope prediction software are IEDB, MHC1. IETB MHC2. Actually, they call it as net MHC1, net MHC2. Okay, so like they have a different names for MHC1 prediction, MHC2 prediction in IETB database. Other than this, we also have immunogenicity. For immunogenicity, we have a separate tool and even toxicity. So likewise, okay, so for each and every parameter assessment, we also have a different tools for T cell epitope prediction. And how about for B cell now? Now we have completed T cell. Your answers are right for T cell. Can we come across the B cell? Even other participants can interact. What are the other? What are the tools for B cell epitope prediction? Yeah, even if you miss some classes, where if you get time, okay, so watch the recorded videos. Okay, even myself, I am checking the recorded videos whether I'm going the correct pace or not. Okay, even yourself, you can develop knowledge on that. B cell epitopes. You remember Chow-Fasman method, immunosurface. I have taken to the IEDB database. Okay, and then I showed you something about B cell epitope, where you have plenty of methods. We have plenty of methods like we have. I think even the Descotope, whatever we discussed, is for the B cell. Okay, you should need a structure of a peptide to get a to predict a best peptide. So Discotope is for the B cell. Actually, for the T cell, we can give MHC1, MHC2 immunogenicity for population coverage, whatever it is. Okay, IEDB is of course a major web resource. Here inside you have B cell epitope like PapiPred, 2.0, PapiPred, Chow-Fasman, Kaplan-Kulzmann flexibility. We have the other algorithms also. Okay, so I think one can go to the ADB website as mentioned, and then you can look through that. Okay, and also one should know what exactly you have to do after predicting the peptides. If you predict the T cell epitope peptides, you have to dock with the corresponding human MHC1 or MHC2 molecules if you are focusing to develop a drug for an immune disease. Okay, if you want to develop a drug for the B cell epitopes, okay, so whatever epitopes you have predicted in B cell epitope prediction, that screened epitopes you have to dock with pathogen recognition receptors. The one such famous pathogen recognition receptors, a group of receptors, or tool-like receptors, TLR. So you remember this slide? I show you the important slide which I showed you on yesterday. This is the important slide I showed you. So we have TLR1 to TLR9. If you go for double-stranded RNA virus, single-stranded RNA virus here. Okay, so they also given what kind of toll-like receptors are can be kept as a targets. Okay, so you can keep this as a targets. If this is about bacteria, then you have to keep this as a target. Okay, so not only it is used for the checking the remedy for cancer, for getting a vaccine for the cancer, even for getting for bacterial viral diseases, you can try with this kind of database and tools present in IEDB. Okay, so I think I recapped something about B-cell epitope prediction. Now let me go to the third chapter, that is about machine learning approaches of drug design. Actually, this is a very large chapter, and I think even the first institute which can even they introduce more about this is IATM. So they came up with a lot of futures, like they came up with a BS kind of a program, BSc programs, for this artificial intelligence and machine learning approaches. Okay, after plus 10 plus 2, okay, some 12, after studying plus 2, they can able to enroll for BSc courses. So almost they're studying for 3 years, and many popular universities, American universities, okay, who have a kind of a branch in India in many places, including Chennai, they are also teaching these kind of a courses. Okay, minimum they're teaching for these courses for a year. Okay, so a lot of concepts are there. So we can't able to complete everything in a single class, isn't it? So I can tell you the basics. Okay, so one should understand the basics before stepping in on acquiring hands-on experience. Okay, even the machine learning itself, we have a lot, but one should understand or even the other thing like the drug design, molecular mechanics, vaccine design, everything you should understand before applying this. Okay, something about me. So, Anna University, I am a PhD supervisor of Anna University. Presently, I am guiding 3 scholars. All the 3 scholars are working on my funded projects. Okay, they're not no need to spend even a single penny for working on the projects. Even I have funded money to work on projects. So I will basically work on drug design, vaccine design, pharmacognosy, and immunotechnology areas, basically focusing on anti-cancer drug discovery and development, anti-diabetic drug discovery and development. Also planning to work on vaccine adjuvants. Okay, so preparing for vaccines. So you are welcome, and even the scholars, those who are working in SVCE, who will be getting a degree from Anna University, also get a stipend of nearly 20,000 per month. Okay, 18,000 to 20,000 per month. So you have an opportunities, like even if fewer scholars or any of them want to learn, so they can learn. Okay, we have an excellent infrastructure. Other than that, we can also prosper with the funding, whatever we say. Now, let me take you to the machine learning. So actually, there are two terms which are interconnected: artificial intelligence, machine learning, bioinformatics, computational biology. We find these terms, see how much closely linked with it, bioinformatics and computational biology. Okay, so let us not discuss about that. Now we discuss about artificial intelligence and machine learning. I think this Venn diagram gives you some kind of an idea, isn't it? So in artificial intelligence, one such subject what we learn is the machine learning. So other than this, we also have another areas to learn. So that we'll be discussing on tomorrow. What are the other areas? Also, we'll see in subsequent slides. What is machine learning, first of all? It is a field of study in artificial intelligence concerned with the development and study of statistical algorithms that can learn from data and generalize to unseen data and thus perform tasks without explicit instructions. You know, whatever programs we work on machine learning or man-made. So each and everything, okay, even if only if you have a training data set, then only the test data set you can predict some values. If you have a sample, if only if the control is having some values that is in the form of a training data set, then your sample value can be determined from the control, or what we call it as training data set. Okay, so this is what general principle we use in machine learning. In machine learning, we use so many kind of programs like Python programming. We can also use R programming. Okay, so these are the popular programmings used nowadays to create various applications. So myself, I am an advanced Java developer 24 years before. Okay, so 23 years before, I am an advanced Java developer. I developed many tools, many software tools for a package. For example, if you take MS Office, Word is a tool, I can tell you. Even Excel is a separate tool. Likewise, for a big package, I developed myself, I created one software. I called it as a PK-CAD. You know, I had Notepad, WordPad features, MS Paint, calculator, and a game. Everything I integrated in the form of a single application. And not only that, even Windows I customized like anything. Like before starting the Windows, it plays the Gayatri Mantram. Okay, a song. It plays a Gayatri Mantram, and even before closing, some kind of another song. Okay, so everything we can customize operating system, we can customize the software. Okay, if you don't like the software present in the operating system, we can use it. So why I'm telling all these things? So this is what, okay, I'm telling you about the machine learning. Okay, if you want to create a tools, okay, if you are giving some set of instructions, then obviously it work accordingly. Okay, so like a creation of tools, generally we call it as a bioinformatics. Then what is machine learning here? The machine learning, if you whatever I explained you initially, okay, comes under bioinformatics, like development of tools. I told I developed some tools and I work with whatever tools I've created. That comes under bioinformatics. But machine learning, how a machine learning differs from this? Here, the machine learning is not just a creating a tool, but a tool you are populated with some data. You are integrating with a database. Okay, you are building a training data set. Okay, with some hundreds of data, and if you build with thousands of data, obviously whatever sample you are going to use to predict the value from the training data set, it is more precise. The best example I can give you is 1000 genome project. Human Genome Project published in year 2003, the final draft published in 2003. But if you see that human genome, with a single human genome, one can't able to uh, diagnose with rare diseases or even few more cancer diseases. With a single genome, with a one single human, how come I can able to find what is the defect gene? So one has to compare with a few population, isn't it? Even there are different ethnic groups. Okay, American groups looks different. Okay, Asian American look different. Okay, and African looks different. So you can find, okay, so even the people with ethic or racial groups will differ with each other. Okay, in the genes and in the phenotype, etc. So one has to try to develop as much number of population in the training data set. Okay, then only the prediction will be more accurate. So one example I provided is a 1000 genome project. Now, not only we have 1000 genome project for the humans, we have for more than 200 species. You know that the 1000 genome project, I think even within a 1 or 2 years, we get more than 300. It's increasing rapidly, the 1000 genomes. Now, not only this, the recent advancement is we are going to get a 1 lakh genome project. How many know about this? 1 lakh genome. Like, so whatever diseases, even if I'm focusing on that disease, it is easier for me, okay, to find the gene and to find a possible solution to how to go for treatment, whether the gene delivery is efficient or we can go for some antibody treatment. So all these things, okay, the decision of developing a remedy, a precision medicine, is based on even the populating the training data set, how much population you are populating in that. You got it? So now let me take you in. Who is the father of machine learning? Arthur Lee Samwell, an American pioneer, is the father of machine learning, is expert in the field of computer gaming and artificial intelligence. See, so he is a field, worked on computer gaming and AI, of course. It's more linked, isn't it, to most of the, even myself, I developed very complex computer games. Whatever I developed, I found it's an artificial intelligence, but no one termed that as AI. He simply called that is a tool, that is a game, that's it. But most of the games automatically work, isn't it? So the games are developed in levels, level 1, level 2, level 3, level 4. Even if it is a Candy Crush or whatever games, okay, you're going to play. Okay, so all this comes under level. So obviously, it works. Obviously, even the chess game, okay, comes rapidly with decision. So it's automatically deciding. See, it's automatically deciding to move each and every coins in the chess. Okay, so all these things are purely an application of AI. Not only you are giving all these data, how to move the coin, how to move my horse, how to move the pawn, how to move the queen. Okay, even in the chess game. Okay, so we have different moves, isn't it? All these moves are fed in the form of a data. So if these coins are moved and what coins are else to be moved, again it's a kind of a set of data. What are the possible chances to move? Okay, everything. Okay, so everything, we call this in the form of a data, and how to automatically work. Okay, this is basically artificial intelligence comes into play the over time period. Now, if you see this machine learning algorithm, okay, we have the types of machine learning. So one is supervised, there is unsupervised. We also have something called as semi-supervised and reinforcement. So totally, I can classify the machine learning algorithm into 4 types. What is supervised learning? So in supervised learning, if you want to know, you have to understand the types. The one number one is classification. The other is regression. You know, all these things, the learning task, whatever they mention, is coming under mining methods, data mining. So if you have a data in the database, if you want to fetch the data, how to fetch the data, how to understand the data, how to infer, how to use the data, everything matters, isn't it? One such example I can tell you, like uh, have demonstrated virtual screening using Autodoc DYRX. You remember that? So how I demonstrated in hands-on experience, I told you to go to the NCBA PubChem. Inside the PubChem, type one compound name. Okay, I typed something like guairevinone. So I got plenty of compounds of guairevinones. Okay, type whatever compounds you know, okay, where you have a series of compounds. If you want to screen that, or even you can try, like if you want to try with the best kind of a drug for SARS-CoV-2, you just type SARS-CoV-2 or COVID-19, even this considered as a keyword. And for that, whatever drugs already showed in the PubChem, I think nearly 1,750 molecules are shown. Okay, so if you don't want to work with all the 1,700 compounds, because your configuration of system is low, and you want to do with only the best drugs, so you can filter the compounds. So you can filter the compounds. How you are filtering the compounds? The filtration is what in the form of a classification. If you are filtering the compounds, molecular weight should be in 350 to 400. You are categorizing in that. You are categorizing the drugs, and you are picking only the drugs based on the particular filter what you have chosen. Now, in this case, is molecular weight. And now, again, if you go for the second filter, if you want to filter the hydrogen bond acceptor should be in the 6 to 8. You are taking it as a second filter for classifying once again. Okay, so this comes under a classification mind. Regression, where basically you have linear regression, or you can go for a multiple kind of a regression. Linear regression, something like if you have y versus x, okay, if you have populations somewhere like this, okay, many populations if you have, I can take in another pen. So if you want to integrate all these points in a single line, I can check which, so if this is connecting all the things, okay, this is the best line I got, okay, a linear line. So now whatever I regression is, or I've taken is the linear regression. For this, I can tell y equals mh plus c. So you can see whatever is close to this line, I can take all this in consideration. Okay, which is very close to this. I can't take this. This is not considered. This is not considered. Okay, even something like a classification, even in regression, you are keeping in the form of a data sets. And similarly, unsupervised. So now we can able to understand. So we are moving in strategic manner. We are keeping a filter and filtering the data and moving in the strategic manner. So for this, you can use these methods: linear regression, logistical regression, random forest, gradient boosted trees, support vector machines, neural networks, for the protein folding. So from the amino acid sequence, okay, if you want to check the not only the structure of a protein, if you want to predict a structure, and after predicting the structure, you want to get a stable structure, then you have to go for protein folding. Of course, the protein structure prediction from Swiss model is also taking care of the protein folding kinetics. Okay, this is something called as protein reaction engineering, biological reaction engineering. So all these things can also be done using this ML support vector machines. Neural networks is popular to find the motifs in a protein. If you want to check which is the functional or structural part of a protein, this is a popular machine learning algorithm we use, neural networks. Decision trees are generally used to diagnose the types of cancer. If a patient, is suffering from liver cancer, suppose think like a patient is suffering from liver cancer. So how a clinician can diagnose initially? First, he has to check what stage of cancer the patient is infected. Okay, so if you want to determine the stage, okay, there are nearly 5 stages of liver cancer. Okay, not only that, if you first he has to confirm whether is it a liver cancer or not, because even the cancers can spread through metastasis. If any organ near to the liver, okay, is succumb to cancer, there are more chances even the neighboring organ, the next organ, liver, can also be getting the cancer, isn't it? So one has to find whether really is facing from liver cancer. Then the next algorithm what we use is whether he is if he is confirmed with liver cancer through 2 or 3 tests, then he has to confirm whether what stage of cancer he is facing at present. Okay, so likewise, okay, so we are going to use a set of algorithms now to confirm the disease and to confirm the stage and to confirm even the severity, or even to confirm the type of drugs to be used for treatment. If because if some patients have history of illness, like for example, if a patient is already suffering from blood pressure, or he is already suffering from asthma, you're already a polio patient suffering from arthritis. And if he is facing from cancer, okay, now he's diagnosed with liver cancer. So whatever drugs he was taking daily, okay, can be taken along with this cancer drugs or not. It's a complicated, isn't it? Taking a decision for multiple diseases, for treating the pleiotropy of diseases. What he can able to do if he is linked with some comorbidity, the decision is most important. So for this, I can suggest you to go for decision trees, of course. Okay, there's a algorithm, and for Bayes, you can use in the application of construction of protein-protein interaction networks, gene-gene interaction networks. I think you learned during the pathway analysis and cytoscape. Okay, so how these networks are constructed? Of course, we should have a knowledge on Bayes theorem. Okay, and now Bayes they basically use the Bayes theorem to construct the networks. Of course, it's a probability distribution function. All these things are probability distribution function. They have their own kind of correlation formulas to develop. A nearest neighbor, I think this is generally used to link different proteins in a species, or link different species by keeping a protein as a reference, as a constant. Okay, if you want to check how a protein is successfully inheriting in the generations, for all these things you can use. Okay, for creation of phylogenetic trees, of course, one can go for the nearest neighbor. Okay, likewise, you have plenty of examples. Okay, even for the unsupervised, but this is not that much structured like supervised. We're not bothered about few concepts. What we bother about the supervised. The base examples or principal component analysis. So the principal component analysis will majorly use it in MD simulation. If you go for molecular dynamic simulation, if you want to check any kind of flexibility, or some kind of flexibility of a protein in particular amino acids, whether if you take a protein, some amino acids structure are not stable. If you want to check that alone, you can go for analyzing through principal component analysis. Okay, so like that. Okay, not checking the entire structure, but you're checking a part of the structure. For this, you can use the uh, unsupervised learning, a kind of a clustering technique used in PCA. Okay, and likewise, you have other algorithms also. Even deep learning models comes under unsupervised learning. Semi-supervised, okay, which is between this and this. Reinforcement learning. Okay, for this, you have these algorithms. The best example you can take is the Monte Carlo tree search. Okay, deep Q learning, temporal difference. Okay, all these things. I think yesterday I told you, na, got an opportunity from one company called Eli Lilly. They are appointing for a director, and the salary they fixed is 1.2 crores per annum. They ask they are asking these kind of skills, whatever I shown, reinforcement learning skills. Okay, there are plenty of companies that give lot of salary, money as a good, decent remuneration if you focus on a particular kind of thing. No need to study supervised, unsupervised reinforcement. Even if you are proficient in reinforcement, you can survive and sustain well when compared to the others. Machine learning in drug discovery. Okay, so we have different steps in drug discovery. How this ML can be used in drug discovery? So now I also give you some, provided you some examples, isn't it? Protein structure prediction for gene finding, diagnosis of disease, protein folding kinetics, linking the proteins of different species. Uh, so many examples I provide. Of course, these are important in drug discovery. Other than that, we also have plenty of examples. Whatever tools I trained from day one, all these tools, of course, rely on the machine learning, isn't it? I'm taking you, I'm first teaching you about the databases, and then I'm teaching you the tools, and I'm telling you how the tools can be integrated with databases, how to fetch the data, how to mine the data, how to, I don't know, how to work on this. All these things, what? The kind of a machine learning. Of course, we are using this machine learning in drug discovery or vaccine discovery or vaccine achievement, whatever components of the drug or components of the vaccine. This is what we are studying from the beginning. Okay, so we can find all this, whatever I explained. Okay, it's can be a drug repurposing. What I explained in the 10th point, drug repurposing. What I explained in SARS-CoV-2. Like I told you, in drug discovery hackathon competition, we won nearly 3.6 lakhs. This is a kind of a drug repurposing we worked on. Okay, for clinical development, we have so many clinical softwares. Okay, where you have to use the clinical data. Not only the data is deposited from the molecular biology experiments, also the data are deposited from the hospitals through clinicians. Clinicians work on the patients. Like if a patient is suffering from the cold, cough, and fever, okay, even the doctor is going to prescribe some drugs, isn't it? They will be taking they consider some 40 to 50 patients, and they take this data, isn't it? Even they can deposit this data in the public domain databases like NCBA. One such popular database is CleanWare. Generally, they won't go to deposit the data like the local health, the health problems like a cold, cough, and fever. They generally deposit the data for cancers. They deposit data for even the multi-drug resistant strains informations, how they are, the drugs are treating these infections. These are most important, isn't it? For the fever and cold, we generally use whatever drugs available in the market. How about the complex diseases like this? How to treat the cancer? How to treat the multi-drug resistance diseases? So even you can find more data getting populated, okay, richly in NCBA cleanware. Like cleanware, you have plenty of even other public domain databases, yeah, even not even in NCBA, even you can find in some other web resources also. Okay, lead discovery, this is something related to how to discover a best drug, okay, out of multiple drugs through visual screening. There is also an example. Okay, and we can see each and everything. We at least we have touched something. Okay, so the cleanware, I think you have to open the database from the NCBA, check the data whatever present, and how we want to annotate. You can annotate accordingly. Okay, so one has to go to the website and you have to browse to the data in the database. Then only get an idea how to work on it. Even you itself can generate your own ideas. Yeah, the applications of machine learning in drug discovery. So unsupervised, supervised, we have plenty of methods and tools. Okay, so one can go to this kind of a URL. Okay, I can paste the URLs at last. Okay, after completion, so you can use the URLs and you can check some information on this. and there's a kind of model. So if you want to work on a particular project, one has to develop a methodology, isn't it? Like even in drug discovery hackathon competition, I told we are used a series of tools. Now, whatever tools I used are the free ones. Even though plenty of commercial software companies came forward to provide free for all the 12,000 participants. Nearly 12,000 participants participated in this, and only 8,000 has submitted properly. And out of 8,000 participants, around 50 participants got some kind of a, not 50, around 75 to 80 people got some kind of a prizes. So out of 80 people, even 12 students, 12 participants are from SVC, Sri Venkateswara College of Finland. Okay, so I'm proud to inform that. And if you want to work on this, if you want to work on this, participant kindly mute. So if I think she's preparing cooking food.
હું દાળ પીળી ન હોય દાળ આમાં લાલ હોય.
So let me take you to the presentation. A basic ML system arrangement. Okay, we use a kind of steps in the flow chart. Okay, and now I take you to the next commonly used machine learning. So one has to find a series, like you have to link with series of tools, okay, to get a solution for a problem statement. That's what I want to mention. So you have to link with the softwares and the methods, and then you have to derive a solution for a problem statement. Okay, the industry people or even the funded project people, they would just want, okay, to develop a solution. So you have to integrate all the tools and you have to get a kind of a solution. Commonly used machine learning software and tool. So this is the best slide, I think, of course. So one, if has to understand about machine learning, one should have a knowledge on the basic softwares. Keras is a very famous one. This is coming under Python deep learning library. Many use Keras in machine learning. Okay, and something which is more popular like Keras is scikit-learn. This is a machine learning tool in Python. Okay, so this is affordable. Anyone can use this tool free. Okay, and you can develop your own applications. Like I think even in LinkedIn, if you're a LinkedIn member, you might have seen if some person is dancing, and obviously whatever creature he has created, even that will dance accordingly. Okay, and he some kind of interpretations initially some 2 or 3 years before during the lockdown, you can find so many people are developing so many kind of artificial intelligence programs. Of course, these are actually they work with this scikit-learn. Okay, and even they are integrated with the keras if there is a need. And TensorFlow, if you have lot of data, big data, not just a small data, you're working on lot of data, better go for this cloud application, TensorFlow. The machine learning linked with the cloud application and open source software library for machine intelligence. And if you want to work on, if you want to create your own chem informatics software, you remember chem informatics software, I demonstrated ACD Chem Sketch. Okay, so in ACD Chem Sketch, you can draw the molecules and you get a 1D smiles format. You can get the 3D structure. All these possibilities are there, isn't it? Even myself, 23 years before, I created my own kind of software to create the components. You can create your own components and you can convert even the formats. Everything, you know, how it's happened. We can work with R. Even I believe even a kind of ASD chemical software can be designed with the R alone, with R programming alone. Even if you see the Discovery Studio Visualizer, this is developed from Perl programming. In Visualizer, if you see the protein, okay, zooming in, zooming out, if you want to find the ligand, how it's bind with the receptor, if you want to work in the form of a movie, you can create that from the Perl scripts. Okay, So even with the common programs like Perl, R, Python, you can find plenty of applications in drug discovery and vaccine discovery. And Vika, Professor Michael Komihas is teaching at least 2 times per year on Vika programming, 3 days program. Okay. This is actually a data mining tool in Java. So he has developed more than 30 tools and 30 databases. So all these tools, okay, can also, if you want to know even how to develop the application, one should have a basic knowledge on Vika, at least how to know use this kind of a tool. And like Vika, there is a tool called MATLAB. Okay, in MATLAB, you also you can create your own tools for your package, for your software. Overview of molecular talking, sampling, scoring, flowchart. What is this actually? Of course, it's a machine learning. I told you now that even myself in my institution, I have developed uh, immunoanalytics software database. How I developed, I just talked some compounds. Okay, nearly I've taken hundreds of compounds and I docked with hundreds of receptors. Whatever compounds I used for docking, I deposited in a server. And all these compound interactions with the receptors, okay, I just used a foreground. I just mined the data using the foreground languages. I used the C# platform through the PHP. I just brought this in a kind of a website. Okay, so in the website, okay, so I can able to mine the data. Like if I give one particular field, I can come to know like which is the best drug for a particular protein receptor. Like this is something related to reverse stock. Okay, this is my kind of an idea, and even if you have your own idea, okay, for any biotech applications, there are plenty of applications to work on. If you have your own idea for all this, all these ideas, even here, whatever shown is this for talking. If you have other kind of thing, if you want to get a kind of a tree-based tree, constructor-based phylogenetic tree, or if you want to go for there are plenty of areas, isn't it? So for this, you can use the machine learning. So one such example I'm showing here is if you're structure-based drug design. If you see the genome sequencing, we have nearly 3 into 10, how much? We have billions of bases in the data in the human genome, isn't it? How much? 3.3 or 3.4 into 10 power 9 bases are there. So that much large population data is there in human genome. If you see the virus, bacteria, the size is less. But if you see Drosophila, I think in Drosophila has more genes when compared to us. Okay, it depends. We don't know. The size of the genome is based on how much genes it have. Okay, so how much bases it have. So the size varies, not based on the size of the animal or okay, it's also based on the size of the genome. So if you have a large amount of data, okay, how to go for finding an appropriate target? Okay, what is the defective gene? So for this, you need a kind of a genome sequencing. Okay, of course, this also here you can find machine learning applications. If you want to determine a 3D structure from a protein, you basically go for crystallizing it. After crystallizing a protein, then only you can go for getting the diffraction patterns through X-ray crystallography, and whatever diffraction patterns you get, like black and white spots, then you go for using the electron density programs. Through the electron density programs, you get a kind of a confirmation, and from this confirmation, finally you get a 3D structure of a protein. Okay, this can be retrieved from the simulation technique. So only through the simulation technique, the electron density uh, data is converted to the 3D structure coordinates in the form of PD. So again, machine learning techniques are used in each and every step. Okay, and even if you have a database of plenty of compounds And if you want to target exactly to the binding site, if you want to check hydrophilic or hydrophobic, and whether is it binding in the binding site regions or not, again, machine learning techniques are used here. Okay, and if you are using a microarrays, and from the gene expression, if you want to come up with a base drug for particular stage of cancer, again, you have to go with the machine learning. So you have plenty of application, each and every step, okay, you have to use a kind of algorithms, okay. And there's an example for breast cancer prediction, machine learning classifiers. So not only these machine learning algorithms are used for treatment, this also used for diagnosing. That's most important. One has to know what stage of cancer is presently facing. Then only, then he can decide the drug. If he is facing less than 50% expression, then he has to choose a drug A. If he is facing more than 50% and it is up to 70%, he has to go to drug B. If he is facing more than 70% expression, then he has to go to drug C. But whatever drug C is going to take, okay, is a strong drug, okay, intense drug, it can also yield some side effect. Something will be there, okay. So one has to decide, even prescribing the drug based on only these stages and other factors, whether he is suffering from other comorbidity of disease or whether he is facing with other illness or not. And I think you remember this I showed you initially, computational methods in drug design, not only for SARS-CoV-2, even for cancer, you will be using all these tools. You know, each and every tool, whatever I showed, okay, in these methods, or of course, using the machine learning algorithms, or using their own machine learning algorithms, and it works according. Drug repurposing, again, you can find plenty of steps you are using the machine learning. So here I just showing you even the SARS-CoV-2. Okay, we have plenty of machine learning algorithms like support vector machine, random forest, artificial neural network, k-nearest neighbor, deep neural network. Okay, so there are plenty of machine learning algorithms used to come up with a kind of a base drug. Not only the drug and vaccines, everything we can come up through using these techniques. One has to get proficient. If I'm an advanced Java developer, why I have to focus on C Sharp? Why I have to focus on ASP, active server pages? Why I have to focus on Microsoft programming? Okay, so you got it. So if you are well versed with the Sun certified application, just focus on that software alone. That's what I can prefer. If you are proficient on Python, you can focus on Python. Don't go with the R. Of course, R is linked with Python. If you want to link like that, you can link. Okay, likewise. Okay, so one has to fix a language and it can develop numerous applications. For support vector machine, again, I can provide you an excellent example is Swiss ADME. In Swiss ADME, remember, if your drug, if you want to classify a drug as a lipophilic drug or hydrophilic drug, generally lipophilic drug is used for targeting brain-related disorders. If you want to target central nervous system or peripheral nervous system, the drug nature should be more lipophilic. Okay. But if you're focusing on other diseases other than the nervous system disorders, one can develop the hydrophilic drugs. There is no need to go for lipophilic. So this I am illustrating with the boiled egg diagram. In a boiled egg, okay, you have an egg yolk in yellow color, which is having high lipid content. The outer, you have albumin, which is having a high protein. You won't have fat. So if your drug, okay, if you try to see the drug is whatever drug you draw in the Swiss ADME or whatever spines format you provide in the Swiss ADME, if you go for clicking predict, you get this kind of an output in the boiled egg diagram. If your drug is lying here, the white region, then you can tell the nature of drug is hydrophilic. If your drug is present in the yellow, you can tell it is a lipophilic. So you can also see plenty of drugs in the data set. In this example, if in NCP PubChem, if you have downloaded nearly some 300 compounds, and all these 300 compounds are lying in this region, okay, you can find this kind of layers. Okay, the hyperplanes separate the classes of drugs. There are two hyperplanes actually. Okay, in one hyperplane you can see most of the red color drugs are there. Might be these red color drugs are lipophilic. And in this hyperplane, you can find hydrophilic. If this is lipophilic, this is hydrophilic. So the hyperplane is splitting, but you also find some kind of lipophilic drugs, but very few, not more, isn't it? So SVM can distinguish the types like lipophilic and hydrophilic in this case. Deep learning, okay. Actually, like if a patient is suffering from cancer and is also facing from other illness, but I don't know what exact drug I want to choose, you know, even we have software applications. All this data. If he's suffering from cancer, this drug has to be treated. But if he's already taking a few drugs for the past few days, then other than drug A, he is prescribed to take drug B. For this, no need to even go to the doctor. There are softwares. The softwares will automatically tell this information. This is what happening in the BPO. For the US people, our Indians are telling what kind of drugs can be used. Okay, by contacting, by telecalling. Everything is happening inside. Of course, they're using a kind of a deep learning algorithms. K nearest neighbors. The best example is getting the drugs information from GMT+5 database. GMT+5 talking trench. I can take you to the GMT+5. I think I didn't explain you earlier. So I'm taking you to the GMT+5. See this how it looks like. I'm just typing in Google like GMT+5. It is taking to the GMT+5 talking.org. So if I'm getting inside this database, okay, so you find nearly 750 million purchasable compounds. Not only that, you also have 230 million purchasable compounds, and you have also analogs, 750 million purchasable compounds. You can search like analogs. Now, generally, if you want to search the compounds, you can search in trenches. Okay, if you want to find the compounds classified, these are the compounds used as a drug. Okay, classified based on the physicochemical properties. So here in the x-axis, you can find molecular weight in Daltons. That is 200 Daltons to 500 Daltons, and even above 500, you have few compounds. Okay, so 200 to 500 and above 500. And here in the y-axis, you find log P, partition coefficient. So likewise, you have the filters, isn't it? So if you want to find this information in 3D, click 3D. Instead of 2D trench, you will be getting now 3D trench. Now, if I want to select specifically molecular weight of 350 and log partition coefficient 2.5, think you want to fetch only the compounds whose molecular weight is 350 and the log P is 2.5. See here, the molecular weight 350 and log partition coefficient 2.5. So you found 48 million compounds. Okay, in the filter, whatever I selected, I found the maximum compounds. So all these 48 million compounds in this particular filter can be downloaded in the form of single data set. One data set. In one data set, you can find all these 48 million compounds. I mean, you find the 3D structures of all the 48 million compounds. So this is a very large database, GMT+5. Not only you have natural compounds, but you also have so many synthetic compounds and analog compounds. When compared to any other chemical compound database, I can tell this is one of the largest database. Even IITM startups are contributing significantly, increasing the population of GMT+5 database. Each and every day, they're depositing 10 to 50 structures. They're making this. Okay, now let me take you to the PPT. Yeah, so how it is happening on? This is working on K nearest neighbor. Okay, K. So each and every neighbor you can find how many compounds are there. Okay, so this is integrated with that kind of algorithm. Random decision forest is best used in QSCR applications. You want to determine the activity for a test data set, you initially have to build a training data set. Okay, so all these things use a random decision forest algorithm. And if you want to diagnose the SARS-CoV-2 infection, not only SARS-CoV-2, even if you take how to diagnose a cancer, one has to know the patient infection level. Then only he can be treated with the appropriate drug. Okay, based on the stage, based on how much infected. So whatever X-ray images the doctor has taken, if you pass it to the software, it in the heat maps you can find the yellow color, red color, etc. If you find more yellow, he is having more infection in lungs. More yellow stands more infection. Then he has to take strong drugs. He can't take mild drugs. And even the dosage level and even the type of drugs, everything concerns to in treatment. Okay, so this is something related to image analysis and acquisition. Okay, for this you can use softwares like 9, TensorFlow, Vika, RapidMed. Of course, again, machine learning approach here comes into play. Okay, not only for the viral infections, but also for the cancer infection. Machine learning. The machine learning, it can, or of we have different methods. Each and every method is used for a certain application in drug discovery or in vaccine discovery. So I told you just about myself, I developed only one database, but my professor has developed a lot. Hard programming for drug candidate selection. So if you see a heme informatics software, you can design your own software, you can create your own data set, and you can also able to do the kind of a drug discovery with your software. So you can see plenty of libraries out there in the R programming to create your own kind of software and to work on virtual screening. Okay, here whatever they are making is a virtual screening. If you want to find which is the best drug, okay, you can also go with this kind of R programming concepts. MATLAB. MATLAB is, of course, a popular software, okay, used for multiple applications. All the branches of engineering also use this kind of MATLAB. Okay, they also have specific tools, plugins to work on the thing. Okay, even for you have a specific module called machine learning in MATLAB. In machine learning, we have subtypes, isn't it? For each and every subtypes also, we have the separate plugins. You can use the plugins and you can work with the machine learning applications in MATLAB. So even the courses are there. We have not only you can work with just the MATLAB. If you know the Python programming, if you want to integrate it, okay, from the Python, you can integrate with TensorFlow, and MATLAB can be integrated with deep learning toolbox. Even these two can be interlinked at last. So almost all the things, R can be linked with Python, Python can be linked with the MATLAB. So one has to get at least provision with one language, then only you can think of to integrate with other applications. Okay, everything is possible. Whatever softwares, whatever you want to make it out, everything you can able to make it up with machine learning. AI, machine learning, and deep learning. Okay, slight differences so that if you want to know more about these three terms, you can also learn some modules. You know, if you have this tool, even learning these modules is free of cost. One can learn these modules, and even after learning, you also get a kind of a certificate if you clear the course. Also, you get a kind of a, but one has to prescribe. You should have a license on. Okay, machine learning is in MATLAB online course. Okay, so I told you now, even you can learn courses from here. So even in machine learning, if you want to learn, you can learn from w3schools.com. Okay, this is the best website. So even the school children can learn. Okay, from the, so it is very easier for you to learn about the machine learning if you start developing from W3 schools. Yeah, I've completed my session. Any doubts you can ask me.
Good morning, good evening, sir. Myself Kalpana Pandya, sir.
From which institution, madam?
I am from SVK, and I am a research scholar.
Good, tell
And
me,
an
madam.
ICMR project I joined here recently. I have good expertise on bioinformatics since my master's, sir. Sir, I have few questions. Can I ask to you?
Yeah, I can.
Sir, first thing is, do I have to learn any programming language to learn this ML methodology?
Yeah, of course, machine learning, or at least you should learn one language. Myself, I learned only C programming. Okay, one day I've gone to the institute and learned C programming. Later, I didn't go to the institute, but I learned self. How many languages I learned? More than 18 to 19 languages I've learned. If you're expert in one language, you can compare with that language and you can last numerous languages. For example, how you can speak the English, even I think you might have learned your mother tongue can be a Hindi or some languages, whatever you learned. You are just comparing the words with the English and you are making fluent, isn't it? Even getting expertized, getting fluent in the programming languages is based on what language you fix. So for machine learning, I prefer you to go with Python or R. Python is the best one. You have so many kind of modules in Python, or you can go with R, or you can go with MATLAB, whatever you have.
So, sir, there are NPTEL courses, so I will learn Python for this. Sir, another question is that as people are saying that it is very easy to get papers, research articles in bioinformatics. That I am from a chemistry background. I completed my master's in pharmaceutical chemistry, sir. But as chemistry demands synthesis and biological work, but we also know in silico work, and that they are telling that we get papers in 3 months. But I am a core bioinformatician. I have worked on enhanced sampling methodology like simulations. The issue is it true to get the papers?
Madam, actually, first you have to get trained in a one language. My suggestion is get enrolled in some Coursera courses. In Coursera, okay, you have plenty of courses in machine learning. So not only they tell some kind of a troubleshooting you should understand. Troubleshooting is very much important. Like if you get in trouble, you should know how to get a solution for that. You should come out of the problem. Okay, I mean you have to get a solution to how to solve. So I suggest you to first initially train with multiple courses. Okay, on what exactly you want to get, what application you are focusing. So even the Coursera courses are delivering certificates freely. Okay, but if you're not getting free, at least you can learn. If you don't want any certificate from that, you learn from that also. So learn from like this. Okay, so you have to focus on a particular application.
Agree, sir. Another doubt, sir, that how like in as you teach MD molecular dynamics in our last course, I attended all the lectures. Sir, how what is the last time, like how long the simulation it will be to get a good impact factor paper with analysis of molecular
Oh,
dynamics?
yeah, you have to integrate with in vitro or in vivo analysis. If you integrate, it can come up to 6 impact factors or 7 impact factor. That's how maximum target. And good, madam, good interaction from you. May I get any other comments from any other participants? Because now the MD simulation is outdated. Okay,
Okay.
it
Sir,
will be.
last question, sir. If you never mind, sir. Last question, sir. Can we get a good funding from different agencies like DBT and other agencies with the in silico work, sir?
Yeah, many people are getting that, but we have to think about to develop an application. Then we will get a more chances. Instead of using the existing tools, if you have an idea to develop a kind of a tool, okay, my professor project, in my professor lab, he is having nearly 12 scholars. All the scholars are just getting the stipend. Okay, so they are getting many of them are getting only from the government organization. So obviously, I can tell you to if you are developing any application, you will have more chances to get funding instead of relying on the existing tools.
Okay. Thank you so much, sir. Thank you, sir.
Any other participants have any other doubts? If no doubts, tomorrow is the last session. Don't forget to attend my last session. If you have any doubts, you can clarify. And I can also show you some tools. Okay, what are the tools and databases developed my in IIT Madras? Okay, you can also check with other institutes and try to acquire few concepts. Okay, so tomorrow is of course the important session for our topic. So thank you very much for your patience and for your interaction, participants.
Thank you, sir.